Hello, I'm Maria Nadestad, and this is the third episode of the OM Genomics Show. I'm going to talk today about applying some principles from the startup world to making bioinformatics software. Now, you may have heard of this book, The Lean Startup by Eric Ries, which is an incredibly influential book in the startup space. It is the foundation of the lean startup revolution, where a lot of tech companies are now building what we call a minimum viable product and going to customers as early as possible, testing that product with them to see if they're interested, if it solves their problems, and what their response is to the product. And then they will make changes to the product based on that feedback, and then go back to customers over and over again with new versions of the product to make sure that they're continuing to improve it. This is far preferable to the old way of doing things, as you might say, which is to have a really big vision for what you're going to create and then build it, the entire vision. Everything you want the product to be, you would build that right away. And then at the very end, you're saying, okay, now it's ready. And then you put it out to customers. And you may find that there's some really fundamental thing that they're just saying, you know what? I never had the problem that this is meant to solve. It just wasn't an issue for me. Or this product is solving it in a completely wrong way. I just wanted something really simple. And it may just be that you've spent maybe years building a really complex product with a lot of APIs and advanced features and all these things, when you could have found out that you were on the wrong track by building something much smaller, a much smaller version of that same product. So that's the idea of the minimum viable product. And I think also in science, this really is an important thing to think about as you're going about building a new computational bioinformatics software application of some kind. If you start with a smaller version of your product, your software, you would have a better chance to see whether you're on the right track, and you can evaluate that yourself if you are part of the target users that you're looking at, or if you have other users who are not including yourself, and you have to go out and show them the minimum viable product and actually get feedback from them and see if that's what they want. Because you may be spending a really long time building something that no one's going to use. And for us, that doesn't translate to lost money directly, although it can mean that you get many fewer citations, you get less interest in publishing the paper, fewer citations down the road from that, fewer grants and so on. So we have a more prolonged process of failure than some startups have, but it's just as important for us to make sure that what we're creating actually resonates with the community if you're putting out a tool that's meant to be used by the community. So I'm going to walk through the process of what I did applying the minimum viable product idea to building Ribbon, which I did while I was at PacBio last summer for kind of an internship during my PhD. And Ribbon was created as a way of dealing with the problem that I had seen in my own research, that it was very hard to see long-read alignments, especially around complex structural variations, and actually be able to interpret whether those alignments support the structural variant or what kind of interesting things are going on. Even if you can't call variants in that region, you can get a better view of it. And that was the idea, to be able to see all of the alignments in a contextual sense, in some kind of intuitive way that makes sense for long reads. So that's what I made Ribbon for. And if you haven't looked at Ribbon already, you can go to GenomeRibbon.com and see what it looks like now. There's another video on that site that just explains how to use it. But in this video, I'm gonna, for the rest of it, walk through my process of creating Ribbon, from prototyping to what the minimum viable product looked like after the first 2 days of development, and then to see how the features have really changed and compounded and how the app has expanded over time in order to deal with the kinds of things that I wanted to do with it once it existed. And I think this is going to be very interesting to anyone who has made an application for themselves or maybe just has used Ribbon before and want to see how the sausage was made. So let's hop into PowerPoint where I can show you some of the screenshots and early mock-ups that I had in Ribbon. Alright, so when I first started making what is now Ribbon, I didn't know what it was going to be called yet. The very first thing I did was to make some pre-project mock-ups. And I didn't know what Ribbon was going to be called yet, so I just called it the Long Read Visualizer. That was all I knew about it so far. And I started by just mocking up in PowerPoint what I wanted it to look like. These are kind of different variants. You can see it was clearly supposed to be, you could see the reference, the contig, and you could visualize how the alignments wrap around different structural variants. So that was kind of the only idea so far. And as I kind of developed this, I looked at, okay, what happens if you have a gene fusion? You might want to see the arrows here maybe represent where the genes are and what direction they're pointing in. You could see now we have 2 different chromosomes and we're looking at how the alignments are going to be visualized. And I wanted to be able to see multiple chromosomes next to each other. So that was kind of a developing idea as well. And so the original idea was that for my long read visualizer, which still didn't have a name, I would make a mini version first, and this was going to be the minimum viable product, is the mini version on the left, where I would have like a link from IGV that turned out to be actually more of a pro feature, but paste in a line from a BAM file or a SAM file, and then I would just view that single reads alignment as a diagram or a dot plot. So that was kind of the minimum viable product idea. And I was then later going to build a separate full version with the full BAM file that you attach, and you do a variants file and you show all these other things, and you should be able to step through the different variants and view reads in those regions. And there were all these features that I wanted to be able to do. So this is the mock-up for the full application where you would be able to visualize genes and variants. You'd be able to see multiple reads, and I wanted a way to show the full alignments for all of the reads, which here was kind of a mini version of what was shown in the detailed view. So where it says reads is the multi-read view now, and the details is a single read view. And you can see I was going to show the ribbon view next to the dot plot. And then you, on the right side, would have a panel where you could upload variants, and you could jump to a certain gene, or you could upload a list of variants and then look at it in that table right there and kind of click on a variant and then jump to that variant. And some of these things came to fruition and others did not. And it's kind of interesting now looking back to the very beginning of the process when I was first developing this and seeing just how much like this ribbon is today and how much it has changed compared to this. Another one, this is kind of the same mock-up, but if you're looking at different genes from different chromosomes, you'd be able to zoom in on both and see those alignments for different regions at the same time. That was a really important idea that definitely did end up in the final ribbon product. Now this is the mock-up for the mini version. And that was what I was going to make first. That's the minimum viable product. In the top left, there's just a giant text field that's a text area in HTML, and you paste lines in there from a SAM file. And then those lines get translated into the table on the right side, where it just shows you basically the same information that was in that SAM file in those lines. And you just like click on one of them, and that would then have an arrow next to it. The green arrow indicates which reader you're looking at now. And then that would blow up on the bottom left, where you see a plot here of just kind of the intuitive alignments there in yellow and purple. And then on the right side there's a dot plot. So I figured you can show the same thing in two different ways, and so I was just going to show them both. And this was the prototype or mock-up for the minimum viable product, which I then went in and created. So then I started creating the minimum viable product, and that was basically just based off of the mock-up. It took me 2 days to get to this point where I could paste in something from a SAM file in that text area at the top. It would become a table on the right side. You can even see the little green arrows over there kind of showing up. And you can tell this looks a lot like the mock-up that I just showed you. And then you would click on one of these, and then it would show up in this plot over here. So the way that this works is that the very top bar here is the reference, and here we're showing whatever chromosomes are available from that SAM file, and then we're breaking up these pieces into just the parts of the chromosome that are relevant. We're kind of zooming in into this second bar, and then the alignments are here. And these alignments I think are real, and then I also made some testing ones that I kind of faked in order to test my own understanding and make sure that the alignments were showing up accurately. So I would essentially make toy examples from some of these lines of the SAM file and look at it in different ways. So this looked a lot like the mock-up. But as I started to think about creating the full application, I actually ended up not making it as a separate one, but building this minimum viable product mini version into the full application. Whereas I was originally going to make them two separate applications, there would be a mini version and a full version, they would be separate. But this one just turned into the other one over time. And so as I started to expand it, I don't have a whole lot of screenshots of what happened in between, but I'm going to show you what it looked like towards the end of August. And this is what it became. The top view here is I ended up calling the multi-read view. And it doesn't look like a bunch of mini versions of this, because you can see there's many more reads than I had bargained for. Originally in the mock-ups, you were showing something like 5 to 10 reads at most, but I actually end up having sometimes 50 reads within view at the same time, and it becomes impossible to actually visualize them like that. So there were some aspects where what I had originally mocked up was actually not realistic given the data. And as you plug in your own data or work with collaborators to put in theirs and look at how you can actually use these applications for actually accomplishing the goals that you wanted the applications to be used for in the first place, you start to realize that some of the things that you had originally thought of are just not useful or relevant or sometimes even feasible to end up making. And along the way, there's also a lot of other features that I added. I hadn't thought of any of the kinds of settings or UI elements here. There's a bunch of different things. One thing is you can change between the ribbon plot and the dot plot. This is what the ribbon plot looks like, and this is what the dot plot looks like. And I actually made them quite similar, where at the beginning there wasn't a whole lot of information in the dot plot. I didn't have any colors or anything. I even have the ability to turn off the colors here still. And these settings are all things that I've wanted to change over time as I plugged in my own data and just tried to use the application to dig into the data in more detail. So I'm going to show you now what the final publishable product look like. And this is what Ribbon looks like right now as of 2017, and also what it looked like when I first put it on the bioRxiv last year. Now you can see that there's genes shown, and this is one of our gene fusions from this cell line that I was studying in my PhD called SKBR3, which is a HER2 amplified breast cancer cell line. And this one is showing one of the well-known gene fusions, which we had found to be a 2-hop gene fusion, which means that these two genes are linked by 2 different variants. And that's shown here by 3 alignments. So you start in one gene over here, and then you can see there's a second alignment to this intermediate region. And this part of MTBP doesn't end up in the final transcript, but then EIF3H over here does. And so you end up with a gene fusion that takes place through 2 different variants. So there's a lot of changes that have happened here over time. I added an information panel as well so that you can see a lot more of what's going on. That way also when you take a picture like this, you can actually figure out which variant was I looking at. It's easier for you to kind of keep that information. So there's a lot of changes that I've added over time, and one of the issues with that was actually that you get this kind of settings creep. I had originally made a panel like this, and I would add in a new setting as it became necessary. Sometimes I want to filter by how many alignments each read has, and sometimes I want to, change different little things. I maybe want to change the color scheme, but there's not a whole lot of features here. And over time, I added more and more features, and I tried to keep them organized with these little headers and so on, but the panel just became huge from doing this. And I was able to make this into a thing that could be collapsed, but it was still kind of confusing to just look at all of those settings at once. So we have this settings creep, where you just more and more settings pile up and on top of each other, and it starts to become unwieldy. So I ended up making tabs here and kind of sorting the settings down into more manageable pieces so that when you're looking at it, it's not as overwhelming. And this really represents how, as you're building a minimum viable product, originally I had planned for no features like this at all. There weren't really any settings that I necessarily knew that I would need. I hadn't necessarily planned to be able to sort the reads vertically. That just wasn't really on my mind at the beginning. But as I started to put in the data and try to figure out what the data is saying from a scientific standpoint, it became necessary to add these features over time. And this is the build, measure, learn cycle kind of from the lean startup, where you start with your minimum viable product, and as you're testing it, you see that, okay, there is something useful here, but we also want to see what are the things that need to be added or changed in order to make it more and more useful as you go from one version to the next. And I kept adding new features. In fact, sometimes I was adding several features a day and just kept looking at the data and, just doing that testing cycle where I would add new features, I would use it again with my data, and I would see where I went from there. What other changes do I need to make in order to really get a good look at this data and understand what it's saying? And so you end up adding all these extra features over time, and that is definitely a good thing. And then you also want to, organize them and consolidate them a little bit. And the code base does expand a lot as you're doing this. But the idea is just to start with something small enough, a minimum viable product, that you can then expand in this way and refine more over time. But you have to have a small enough minimum viable product that you're able to test it. So this fits the definition of a minimum viable product very well, because it allows me to test whether the visualization that I had planned to do here, which is shown here in blue, if this one core piece of the ribbon application, if that was actually going to be a useful way of looking at these reads, and would it give me any extra understanding that I wouldn't get from reading the SAM file by eye and drawing it on paper, or plugging it into IGV and looking at these 2 regions completely separately. So chromosome 17 and chromosome X, you wouldn't be able to see all this in one view, and you can in Ribbon. And that was the core idea, was that you would be able to visualize multiple regions at once and see all of the alignments for a single long read in one view to get the full context of what are all of the alignments for this particular read. See them all in one view. That was the core idea. And this was the smallest possible application I could make in order to test that. You have to have some kind of an input. Even the table is not even necessarily necessary, because you could put in a single line from the SAM file and still test it like that. But the table wasn't very difficult compared to making this visualization work, especially with having multiple scales up here. And that was the challenge. So this whole thing, I think including the mock-ups, took me 2 days of full-time work to just produce this. But once I had it, I was a lot more motivated to add extra features. Immediately, I was just like, it would be really nice if I could, see all of these reads together and not just individually. So that's when the multi-read view came about. And I also wanted to plug in other things than SAM files. Maybe I had coordinate files from Mummer, from de novo assembly alignments, that I also wanted to visualize in the same way, and I start to realize that you can use different kinds of data with this application. I also wanted to read in BAM files, which turned out to be a little bit more difficult, but there was a library available for that from BAM-IO-Bio. And putting all of these things together did take a long time, but creating a minimum viable product in just 2 days, just to get to this point, was by far like the 2 most productive days of the whole experience, because it allowed me to test the basic ideas and see whether the visualization was actually useful. So building a minimum viable product allows you to test the core idea of what you're trying to accomplish and just to see whether that thing is useful. If that creates interest in your customers or is useful for yourself, for your own data, if you're building this for your own research, then that is something that you want to move forward with. If I had built this and I had thought, there's no significant impact of this beyond just using IGV, or I can easily draw this on paper in 2 seconds, why would I want to plug it into an application to do it for me? Then I would have scrapped the project, or thought of something else to do, or thought of some more important, more core way of adding value. You really want to be careful to create the smallest minimum viable product you can. And it's very motivating to have that minimum viable product that you can then expand on. Because once I'm at this point, there's so many things that I can think, I'd really like to add this. I'd really like to be able to see multiple reads. I really want to read in BAM files. I really want to look at Mummer coordinates. I really want to be able to hover over each alignment and see how long it is. I really want to be able to zoom in on a particular genomic region and look at one variant and see what are all the reads around this variant. You get all these ideas once you start just making a minimum viable product and testing it with your data. So this is what I would highly recommend for anyone who's starting to create their own computational methods, bioinformatics software, especially if it's also going to be used by other people, to just start with a minimum viable product and test it and build it up from there. So that's how I made Ribbon, which is at GenomeRibbon.com, and you can look up the code from there too. There's a link to GitHub under the info tab. And I encourage you to go and check that out and to try to think about applying some of these ideas, like building a minimum viable product for your own applications, especially if you're building one of your first computational tools in bioinformatics software. Thanks for watching this 3rd episode of the OM Genomics Show. If you have any comments, feel free to leave them below. And if you have any questions at all about this, I'd be happy to answer them in the comments as well. Please do hop over to omgenomics.com/subscribe to sign up for weekly email updates on everything I'm doing, including some of the new visualization tools that I'll be building, as well as more videos like this when they come out every Friday. So I hope to see you over on the email list, and have a really great day. Thanks, bye.